My name is Amy Mullen. I'm the Associate Chair for the graduate programs in the chemistry, biochemistry at the University of Maryland, College Park. And welcome to our virtual information session. I'm going to give you about a 10-minute overview of our department and the research going on. And then I'm going to hand it over to Doug Julin, who's the program director of the biochemistry program, and then Efren Rodriguez, who's program director of the chemistry program. And then we're going to turn over our discussion to a panel of faculty researchers who will present overviews of their research programs. And then we'll open it up for questions from students. Then at 2 p.m., our graduate students are going to take over and lead us a panel on student life in the chem and biochem programs. And you can find out from the source what it's like to be a graduate student in our department. Okay, so what do graduate students do here? They take courses, and the set of courses are tailored to students' individual interests. They receive a foundation for investigation in theoretical and experimental studies, and they lead and design independent research projects in collaboration with their research mentors, and then communicate their successes at conferences and through publications. So there's a wide range of research activity in our department, and the research ranges from traditional topics to interdisciplinary topics, and these are listed here. We have 3 research themes that are department-wide, and they fall under the general categories of multidimensional biomolecular science, or BioD4. And this comprises research in bioanalytic, biophysical, chemical biology, and synthetic research to investigate biomolecular function. We also have another research theme in multi-scale molecular science. So these are measurement and simulations of processes from the scale of the atom out to the cosmos. This includes laser studies, advanced materials, imaging, simulations, quantum chemistry, and artificial intelligence. Our third major theme is chemical innovation for emergent and sustainable materials. Researchers under this theme use synthetic chemistry, quantum materials chemistry, and chemistry focused on energy and sustainability. We have research mentorship from over 30 faculty members, and these are all listed here, and you can go to our website to learn more. We also have first-class research facilities and instrumentation that are available for research projects. Okay, the University of Maryland is located very close to the District of Columbia, our nation's capital. And we're also very close to a number of other institutions, research institutions, universities, colleges, as well as federal agencies. So there's lots of collaborations that we're involved with in this area and beyond. I wanted to say a few words about the application process. So our deadline for graduate applications is January 14th. What you submit are the following things: a statement of purpose. This is one of the most important parts of your application. This is where you tell us what your goals are for graduate school and what research labs interest you most. Be yourself and be as specific as possible. We want to know what you want to do when you come here. Send us your transcript or transcripts from college. We want 3 letters of recommendation. It's best if they come from people in the sciences. Your CV or resume. If you're an international student, we need an English fluency exam score. You can send us any publications if you have them, not required. And this year we're waiving the requirement of GRE scores. If you have them, you can send them, but you don't have to. And then the review process consists of a faculty committee we perform a holistic review of the applications, which means we look at the entire package that you send to us. So we don't have specific cutoffs for admission. So we make offers of admission between January and February. We'll host a Visit Maryland Day on March 12th. It may be virtual, depending on the health situation of our country. And then students will respond with their answers to offers by April 15th. Okay, so I'm going to turn this over now to Doug Julin.
There's a question in the chat.
Oh, okay.
The question in the chat is, does the transcript have to be an official transcript?
Yes.
Maybe I'll ask if there are any other questions before I begin.
I mean, what I can say about the transcript is that to be admitted to the university formally, we need an official transcript. If you can't get an official transcript, you can send us an unofficial one, but we can't formally admit you to the program until we get the official one. Okay. Doug, do you want to go ahead?
Yes, I'll go ahead. So good afternoon, everyone. Thank you all for coming. I'm the director of the Biochemistry Graduate Program, which is part of this larger department, and I will tell you about our program. Right now, there are 35 graduate students who are working towards the PhD in biochemistry, and a PhD is about research. So here I've listed a number of research areas and keywords that apply to most of the faculty who do biochemistry in our department and in our program. And basically, almost any biochemist studies the structure and or function of biomolecules, including in our program, proteins, enzymes, glycoproteins, and of course, DNA and RNA, in various ways, and asking a wide variety of questions about these biomolecules. Several faculty use high resolution, high field NMR or X-ray crystallography to study the structures of each of these types of molecules. And then here below, I've listed some other sort of keywords that describe the research of various faculty, and to give you a sense of the breadth of research that goes on in our program. Here I've listed faculty. So these are faculty up here. There are 13 faculty in our department. These are the faculty who do biochemistry, broadly defined. They are the faculty who teach the biochemistry courses, and they interact, I think, most directly with the biochemistry graduate students. I point out, you should note that 2 of these faculty are off campus. They are situated at the Institute for Bioscience and Biotechnology Research, IBBR, which is a research branch of the University of Maryland. I've also listed a couple of faculty who are in other departments, but they have affiliate appointments in our department. And they also, their research interests very much overlap with biochemistry. Now, if you want to know more about what these individuals do, go to this website that's given down here, and you will find more information about each of these faculty members. And you will also find a more detailed description of the research that each of them does. Now, in addition to research, of course, there's other aspects of professional preparation that are an important part of a graduate program. There's coursework. So we offer coursework in basic biochemistry, also coursework that addresses these other skills that are very important for your later career: oral presentations, research ethics, and writing. Most of the students finish the main coursework in their first 3 semesters. And after that, the focus is almost entirely on research. We have a weekly biochemistry seminar in which Scientists come from other institutions, mostly to tell about their research. Our students attend national and in some cases international conferences. We host career planning events. And I should point out that graduate students in our department receive full financial support. That is a stipend either as a research assistant or as a teaching assistant. Finally, I'd like to just comment on what our students do after they graduate. Most of our students immediately after graduation take a postdoctoral position. And this is a research position in an academic lab, but also it could be at a company, at an industrial company, could be at a national lab such as the National Institutes of Health. Postdoctoral positions usually last 2 to 4 years. And after that, graduates move on to more permanent careers. And I've listed some of the career paths that our students have taken, our graduates have taken. Some of our graduates have positions in academia, but many of our graduates have positions in industry, in the biotechnology industry or pharmaceutical industry. Some of these are positions involving laboratory research, but also some of our graduates have positions in sales, management, and so on. Now, situated as we are just outside Washington, D.C., we're very close to the headquarters of many government agencies, and a number of our graduates also have positions at the NIH, the Food and Drug Administration, the Patent and Trademark Office, and others. And again, these positions can be research positions, but they can also be more administrative types of positions. So a PhD prepares you for a very wide range of career paths, and our students have taken many of those different paths. All right, so my last slide is if you want more information, here's my contact information. Again, I'm the director of the graduate program. I also will refer you to the graduate program office headed by Diane Cantor, who's the coordinator of our graduate programs. And here's an email address for the graduate program office. And finally, here are the two websites that I listed on my first slide. And you can go to those websites to get more information about our programs and our department as well. Thank you very much.
Thank you, Doug. Now we're going to switch over to Efrain Rodriguez. He'll tell us about the chemistry graduate program.
Okay. Thank you, Amy. All right, welcome everybody to our info session. I hope that you apply to our program, and I hope that we see you next year doing exciting research in chemistry. So I am Efrain Rodriguez. I am the director of the chemistry graduate program. And if you want to contact me afterwards, you can always email me at efrain@umd.edu. Or as Dr. Julian was just saying, you can also contact Diane Cantor in the graduate office. So in chemistry, you can come in and partake in many different research areas. Dr. Mullen gave a nice overview of all the different research directions our department is engaged in. This is just a list of both sort of the traditional areas highlighted in red and sort of the interdisciplinary areas within chemistry that you can do research in. So for example, we can have materials, photochemistry, quantum science, and so forth. Also, I would like to say that, you know, you can join the groups of all the faculty in chemistry, or you can also look at faculty that are listed as affiliate in our department. And some of our chemistry students also look into the groups, the PIs in biochemistry as well. Let me tell you specifically about what it looks like to be a first-year graduate student in chemistry. So when you get here, really what we want to do is get you on the path to a PhD in chemistry. We want you to join a research group and become the world's expert in your project. In the end of the day, you will be the sole author of your dissertation. And so you will actually know more than your PI in your field of expertise. We expect you to come here and now start to create knowledge as well. You present your results at conferences, meetings. These can take many forms, whether it's the large national meeting of the American Chemical Society with thousands of chemists, or it can be a more intimate, smaller meeting like the Gordon Research Conference. We expect you to publish your results in chemistry journals or physics journals. A lot of us actually publish in related fields like materials science, for example. Um, and of course, your first year, you'll be also engaged in coursework and academic work. So you'll take a look at the courses that are suited to your interest in research. So we'll guide you and advise you when you first get here on course selection. You'll be taking Chemistry 611, Chem 611, which is a way to sort of socialize the new incoming class so that you get to meet your peers, your cohort, also learn about certain subjects like writing, ethics, responsible conduct of research, and so forth. We really try to integrate our incoming class with the entire department and our program. Another way to do that is actually by attending seminars. Dr. Julin mentioned the weekly biochemistry seminar that happens on Tuesday, but we also have one on Wednesdays, which is physical and analytical. And then we have one on Thursdays, this inorganic, organic. Now, these are the traditional the areas in chemistry, but as I said, we don't have any formal divisions. These are just titles to help us roughly know the type of research that will be presented at those seminars. So it's a good way to connect with other group members, other groups in the department, and to see PIs as well. And of course, to see outside speakers, since those are predominantly who come present in our seminars. So our path to PhD. You will be required to take 21 credits by your fourth semester and be in good academic standing. Present a literature seminar in year 2. This takes the place of oral exams. And then later, you'll also do a so-called candidacy exam, which is an oral exam with a written document where you propose your research. Once you're done with that, then you're on to the second part of your graduate career, which is to do original research, publish your work, present at conferences, and write your thesis and defend it. So we're looking for you to advance candidacy by the end of the 4th semester or 5th semester and complete your degree by the end of the 6th year or earlier. So I'm not going to get so much into the weeds of what will happen the first year. I'm just giving you an overview that in your first semester, you're free to look around and interview with as many PIs as you like, because you're under no pressure to join a group until December 1st. Okay, so before then, you can actually talk with many groups, go to their group meetings, for example, meet with their students and their postdocs. November 1st is the earliest date that you can join a group. And so that's what we're looking for you to do once you get here on campus next fall. And as I said, we have the seminars on Wednesdays and Thursdays, always at 11 a.m. So that makes it easy to work into your schedule. Rather than going into more details, I'm just going to say that your goal within the next 2 years when getting onto Maryland is to advance to candidacy. And if you want more information on coursework, how you should think about your literature seminars and so on, I am always happy to answer those questions through email. And after this, we'll talk with panel of faculty. I'll be on the panel myself, and I'm happy to share with you where our students go to after getting a PhD. I'll show you some of that information.
Okay,
panel.
is that it, Efren?
Yeah.
So Before we go on, are there any questions about sort of the format of the two graduate programs? And you can either just speak out or raise your hand or put it in chat.
Yes, I have a question.
Yes.
If for some reason the candidate doesn't want to continue with the PhD, what's the process for, or is there a process for the master's?
Certainly. So
Yep.
the requirements for a master's degree are slightly different than the PhD, but it's basically coursework and then either a literature paper and presentation, or literature paper, or lab-based research. So those are if you decide that you don't want to get the PhD, you need a 3.0 average and a certain number of courses, and you do need a research course. But that's a good discussion to have with yourself and with your research advisor. If it turns out it's not what you think it was, you do not have to continue to finish it, and you can leave with a master's.
One thing I forgot to mention for the chemistry program is that you don't have to do rotations in chemistry. So it's just about meeting with PIs and interviewing with them. So there's no like formal rotations in chemistry, unlike biochemistry.
Yeah, I didn't mention that in the biochemistry program, we do have lab rotations, which I think are fairly traditional in biochemistry programs. So you do 3 lab rotations with 3 different faculty, and that helps you to get a feel for what the faculty member is like, what their lab environment is like, and helps you to choose the advisor that you're going to work with for the next 4+ years.
Yeah, and in chemistry, you can arrange to do a lab rotation, but it's more entrepreneurial. You set it up with the PI that you're interested in, and spend a month going to group meetings, spend some time in the lab, shadow a grad student. It's a good idea to find out what the feeling of the lab is and how the research is done. What's the style? Does it appeal to you? Does it seem interesting?
Yep. And we've even offered opportunities in the past. If you're really gung-ho about coming here, is in the summer before the fall term to come in and do a little bit of lab work, just get a sense of what it means to be a graduate student.
Okay, any other questions? Okay, we'll have time for questions after the faculty panel as well, so you can keep those in mind. So now we're gonna switch over to the faculty panel. We have 9 faculty presenters, and this is just a small sample of the research that's going on. We had to limit it to 9 because we only have so much time. And I've asked the faculty to speak for 2 to 3 minutes. So if they go over, join me in yanking them off the stage. Okay, so we're gonna ask Miles Poulin to go first.
All right, sounds great. Here we go. So yeah, just want to give you a quick overview of my research program. A lot of, let me see.
That looks good.
Okay, you can see everything all right?
Yeah.
All right, so my research program is really at the interface of chemistry and biochemistry, and we're interested in using both chemical and biochemical approaches to look at the role carbohydrates play during bacterial biofilm formation. So a lot of people know bacteria as single-celled organisms that are sort of these free-floating organisms, the planktonic form of the bacteria. But in reality, most bacterial species spend a significant portion of their life cycle in multicellular communities that are encased within a self-produced matrix of biomolecules that allow them to form complex 3-dimensional microstructures. We're interested in the role carbohydrates play during this process, and we really have 3 main research themes in my group, which are to use chemical approaches to study the mechanism of enzymes that are involved in assembling and disassembling these carbohydrates. We are developing tools to allow for engineering of enzymes to catalyze the breakdown of these polysaccharides for the purposes of developing biofilm dispersal agents. And we're also developing physical organic chemistry tools to study the transition state and mechanism of some of these enzymes in order to help design inhibitors. So those are the three main research themes in my group. If you're interested in more information, you can always reach out to me. I put my email address here. The more information about the lab can be found at this website, and I will pass it on to our next faculty speaker.
Okay, thank you, Miles. Efren, you're gonna go next.
Hello, everyone. So you just heard from me on the graduate program. I run a group that's dedicated to research in functional inorganic materials. These are extended inorganic solids. And so these are the three different areas in my group that are currently under investigation. So we have on the right side, we're looking at layered metal chalcogenides for quantum science. We call these quantum materials nowadays. It's a new phrase and style. And so in particular, we look at superconductors and magnetism. And this is work funded by the National Science Foundation. More recently, we've gotten a grant from the US Department of Energy on looking at metal oxides, again, for magnetism. But really, this was more focused, this is more like a technique focused research where we use neutrons to scatter off these magnetic materials. Neutrons themselves have a magnetic moment, and so you can do crystallography with neutron scattering. A lot of that is done nearby at a lab at the National Institute of Standards and Technology and also at Oak Ridge National Labs. We also look at the reactivity of metal oxides, transition metal oxides that are redox active for energy conversion and catalysis. And so this work more recently has been funded by the Defense Threat Reduction Agency and the Army. This is known as DTRA. And here we're studying them now for the adsorption and defeat of chemical warfare agents like sarin or VX gas. But we've studied them for other types of surface reactions as well. That's just the one that right now is currently funded. And you'll be hearing actually from one of my students, Matt Leonard, who is on this third project. I just thought I would give you some recent papers in each one of these three. For example, on the top left is a paper that we published this year, and it doesn't seem like it has much to do with layered metal chalcogenides, but in fact, it's how we synthesize them. We synthesize them in water under what's called hydrothermal conditions. And we found this very interesting isotope effect between water and heavy water in the synthesis of these materials. In the neutron scattering area, we're studying these particular metal phosphates called olivines and trying to distinguish sort of subtle features in their magnetic order. And then finally, this paper on what's called chemical looping, where we react these metal oxides with methane to see if we can sort of use it sort of as a pseudo catalyst that came out last year. This is my group. Currently 7 graduate students, 2 postdocs, and this number fluctuates, number of undergraduate interns in my lab. This is my daughter, not an undergraduate intern. But these are the past members of my lab. And just to give you a flavor of where they go, national labs such as Argonne. Joe is actually at Argonne and Northwestern University in joint appointment there. Naval Academy, also industry, Rishvi's at Intel. Dan is at Ford Motor Company, and Marcus is at currently at the National Institute of Standards and Technology nearby. So that I'll end my talk.
Okay, thank you, Efren. Now we're going to go to Lyle Isaac.
Okay. Hi, I'm Lyle Isaacs. I'm an organic chemist. I run a research group that's very interested in supramolecular chemistry. The molecules that we're most fascinated with are a class of molecules known as cucurbituril molecular containers. This is a picture of my research team. This is a pandemic picture, so we're a little bit different than what you see there, but that's about my normal size of group. I'm very interested in containers. So in everyday life, containers are very important for keeping your soda from going flat, for shipping presents to your friends, for organizing your stuff. And it turns out that molecules need containers too in order to improve their properties. For the past 20 years or so, I've been studying these molecules known as cucurbitryls, which are able to hold on to molecules very strongly inside their cavity. We've done a tremendous amount of work designing and synthesizing and evaluating the properties of a variety of derivatives of the cucurbitryls, where we put functional groups in various positions. About 10 years ago, we started working on biomedical applications of cucurbitryls. And I show you here an example of a molecule that we call Motor 1, after my student Motor, who made it. And we found that this molecule has a wide variety of important applications. For example, a lot of insoluble drug molecules cannot be formulated to give to humans. We can formulate them by having them dragged into solution inside our containers. We can, for instance, increase the solubility of Taxol by 3,000-fold by this technique. For drug molecules that bind very strongly to our containers, we can perform a different class of applications, and that's known as reversal applications. In anesthesiologists administer a compound known as rocuronium hundreds of millions of times per year to patients. But patients, when they're done with surgery, they can still feel the effects of rocuronium. And so we've developed a compound known as Motor 2 that has the ability to reverse those effects of rocuronium in vivo in rats. The other class of compounds that you'll be very, very interested in sequestering in vivo are drugs of abuse. So our containers are highly complementary to alkyl ammonium ions. Ammonium ions in biological systems, probably the most prominent ones are drugs of abuse, things like methamphetamine, cocaine, and fentanyl. You may know about naloxone as a way to save people from dying due to opioid overdose, but methamphetamine and cocaine are not opioids. What we've done is we take our containers, we administer them to animals. You can see here animals walking around the outside of a box when they're not on meth. When we give them meth, they run around like crazy. When we give them our compound either before or after meth, you can see we can control their hyperlocomotion. And so this compound is currently in the process of going towards clinical development. So we're very excited about that. The past couple of years, we've studied a few new scaffolds known as pillar arenes, which also have excellent molecular container properties. And we're very interested in metal organic polyhedra that we've now decorated with cucurbitryl molecules. Students in my group develop core skills in supramolecular chemistry, molecular modeling and design. We perform a lot of organic synthesis. You may think these molecules look torturous, and they are. You become, of course, well-versed in things like NMR, calorimetry, UV vis, fluorescence. And importantly, the students, even though they don't perform the biological experiments themselves, they become well-versed in the language of the biological collaborators. So with that, I'll stop. Thank you.
Thank you, Lyle. Now we're going to hear from Leah Dodson.
All right. Thank you, Dr. Mullen. So I am Leah Dodson. I'm a second-year assistant professor here, and I'm getting my group started. What we're really interested in doing is using experimental techniques that are developed in physical chemistry labs or even in physics labs to study systems that are of astrophysical interest. So we're a laboratory astrochemistry group. And the key factor that we're interested in controlling for when we study laboratory astrophysics is the temperature and how temperature contributes to the differences in chemical reactions that we see in space or on planetary atmospheres. So there's two main interests in my group. The first is circumstellar chemistry, the chemical reactions that are happening in evolved stars that are spewing gases out as they die. We're interested in how you can link metal chemistry to eventual formation of planets in future iterations of the stellar evolution. So my group is developing a new tool to study ion molecule reactions involving metal ions. And we're using some of the techniques that we're stealing from the physics departments, such as the cryogenic buffer gas beam source, which is a great, easily incorporated new source of molecular beams, as well as some established technology that has really come to the forefront in mass spectrometry communities, such as the cryogenic ion trap. So we're going to be studying this particular reaction, magnesium ions plus cyanoacetylene molecules, such as or cyanopolyene molecules such as the cyanoacetylene reaction. And this is important in circumstellar chemistry, like I mentioned. The second research thrust is for planetary atmospheres. It's a really exciting time right now, especially in the field of astrochemistry, as we're discovering new molecules on new planets and celestial bodies. You may have heard something a little bit about phosphine recently in the news. So we're interested in providing chemical explanations for the chemistry that's occurring on planetary atmospheres. And to do that, we are using matrix isolation techniques and spectroscopy, including infrared spectroscopy. And we're using that to study mechanisms such as the one shown here, where you have an isomerization reaction that can be catalyzed in the presence of a hydrogen bond donor. My group, you can follow us on Twitter. Please contact me by email. I'm happy to talk more with you and hope to see your applications come through this fall.
Okay, thanks, Leah. Our next speaker is Dorothy Beckett.
Okay, my name is Dorothy Beckett, and I'm a biochemist, and actually I'm more of a biophysical chemist. So, and what we study is biological regulation, and we study it at multiple levels. So whenever you talk about biological regulation, you want to talk about allostery. So we use one system, a reference system for these studies. It's called the Biotin regulatory system, and I just went out. Okay. And the Biotin regulatory system is it's a molecular switch where you have a single protein that switches from metabolism to transcription repression. And it's a single protein that's capable of doing this. And we study this at multiple levels. And at the heart of this ability to switch is something called allostery. And allostery is probably the most important regulatory process in biology. But although we've known about it for over a century, we still don't understand it at a molecular level. So part of our work is devoted to understanding how a signal from one side of a protein is transmitted over many angstroms to the other side, what is going on internally in the protein. Second feature of the work is this is a regulatory switch that is actually found throughout the all bacterial kingdoms. But actually, many bacteria can only serve as enzymes, or their protein that is central to this can only function as an enzyme. It doesn't regulate transcription, and yet the architecture is nearly identical. So then the question you have is, how is it that you take a single enzymatic function and somehow you add on the ability to allosterically activate transcription? You have to pick up a DNA binding domain, but you also have to change what is going on internally in the protein. So this is more of an evolutionary study. How do proteins acquire new functions? And then we go to a higher level, which is to ask, if for any system, regulatory system, there's a biological output. In this case, the output switches from metabolism to transcription repression. And there are multiple interactions that contribute to that. And so what we focus on is, how is it that each of these different interactions regulate or tune the output? And what is shown right here is that we can tune the transcription repression function of this system by just making single amino acid substitutions. And it's very, very sensitive. The transcription output, and actually it's biotin that regulates that output, is highly sensitive to single amino acid replacements. So we use a wide range of methods in our work. We use molecular genetics, standard biochemical purification and characterization, biophysical measurements, including ITC and analytical ultracentrifugation, fluorescence, rapid kinetics. And we're doing a lot of computation and bioinformatics as related to these two problems, some of that in collaboration. So people who go through my lab end up with a very good preparation, very strong preparation in the whole range of techniques that are important for being a very well-trained biophysical chemist, and people from my lab end up going into biotechnology companies for the most part. So if you have any questions, you can just send me an email at this address. And thanks for listening.
Thanks, Dorothy. Our next speaker is Pratush Tiwari.
Well, it's always a tough call to go after Dorothy, but I will try. So I hope you can see my screen.
Yes.
Great, wonderful. So my name is Pratush Tiwari. I'm a relatively new professor at University of Maryland, even though time flies. I'm in my 4th year of tenure track. And I'm jointly appointed in chemistry and biochemistry always, and also this institute called Institute for Physical Science and Technology, which allows my group to really do interdisciplinary research grounded in physical chemistry. So the dream of my group is to really develop a perspective to go straight from atoms to mechanisms. This idea that everything can in the end be described in terms of just jiggling and wiggling of atoms. If you know what is the force through which two atoms talk to each other, you can solve Newton's law of motion and do it on supercomputers and tell everything that is to be known about, for example, how does a crystal nucleate over here, or how does a little ligand such as benzene leave lysozyme, or how do more complicated biomolecules such as riboswitch recognize and interact with ligands. So in order to do all these, our workhorse is something called molecular dynamics simulations, which, as I said, is just solving Newton's law of motion. We do it on a bunch of supercomputers, graphical processing units. However, the problem in these simulations is that they are quite slow in the sense that if you run it for 2 months, so think about running a computer code on 100 computers at the same time without touching those computers for 2 months. If you do that, then maybe you will get to 10 microseconds. Not a lot of biology or chemistry happens at the 10 microsecond time scale. So what we do is to develop special methods which push this time scale of molecular dynamics simulations. And we publish these methods as open source codes that are then used by scientists across the group, which is extremely exciting because if you have ever written a piece of code, you know how easy it is to make a mistake. And when the whole world starts to use it, it's like opening up your code to the whole world. So, and our group is very interdisciplinary. We have students at last count from 5 different departments, including chemistry, biophysics, physics, applied math. We get a lot of undergraduate students, really hardworking and talented undergraduate students from biochemistry and chemistry and also computer science. So if you join my group, you get to interact with not just students in chemistry, but really top-class students from across departments at the University of Maryland. And definitely you will get to hang out a lot with my dog. His name is Pakoda. And my lab is a new lab, so not a lot of our people have been placed many places, although they are headed out to good graduate school. And my first postdoc is negotiating his first tenure track position right now, so that's very exciting. These are some of the publications we have had in the last 3 years since inception. We publish in traditional chemistry journals, such as Journal of Physical Chemistry, of which Amy was the editor for many years, and also in hardcore structural biology journals, but also more broad journals. And our funding comes from all over the place, again, reflecting the nature of our research, from Petroleum Research Fund to Basic Sciences to National Cancer Institute. And as Leah mentioned, just like Leah, I'm also on Twitter. You can come and tweet at me there or send me an email and I will be happy to answer any questions.
Okay, thank you, Pratoush. Our next speaker is John Forkas.
All right, I did a narrated PowerPoint, so I'll stay on time and speak intelligibly. Welcome to this quick video tour of research in the Forkas Group. Our research focus is on the use of ultrafast lasers and nonlinear optical techniques to probe, control, and fashion condensed matter. Although the majority of our work is experimental, we also do theory on light-matter interactions and perform simulations that address issues related to the structure and dynamics of liquids. The people in the Forkas Group come from a wide variety of scientific backgrounds. No prior laser experience is necessary. We are looking for dedicated and enthusiastic researchers who like to work collaboratively. Members of the group not only work with one another, but also with researchers in our department, other departments at the University of Maryland, and around the world, as well as with researchers in industry and at national labs. Ultrafast laser spectroscopy uses lasers that have pulse lengths up to hundreds of millions of a billionth of a second long. These ultra-short pulse lengths allow us to monitor chemical processes in real time and to drive nonlinear processes at low average laser powers. One major element of our research is the use of ultrafast nonlinear optical spectroscopic techniques to study the properties of liquids. One such technique is optical current effect spectroscopy, which allows us to monitor the orientational dynamics of liquids. We are interested in how these dynamics depend on thermodynamic parameters, such as temperature and density, in bulk liquids, as well as how they depend upon confinement on the scale of a few molecular diameters. Another technique that we use is called vibrational some-frequency generation spectroscopy. This method allows us to collect vibrational spectra of molecules at interfaces, even when the population of bulk liquid molecules is orders of magnitude greater. We use this technique to probe the organization and dynamics of interfacial liquid molecules in technologically relevant circumstances. Yet another nonlinear optical technique that is used routinely in our group is multi-photon absorption. This method allows us to localize absorption within the focal volume of a tightly focused beam of ultrafast laser pulses. Consequently, we can perform three-dimensional imaging of a wide range of interesting luminescent systems. Multi-photon absorption can also drive local photochemical and photophysical effects. One example is the cross-linking of polymers. Here we show examples of 3D structures that we have created using this technique. We are also pursuing many applications of multi-photon absorption polymerization. Shown here are some examples in cellular biophysics. Parallel nanoridges separated by a distance that is much smaller than a cell diameter lead to directed migration, as shown on the left. The mechanism for this phenomenon involves the preferential nucleation and guidance of ways of actin polymerization, as shown on the right. If you have seen things here that interest you, please view our video at the URL shown here, or send me email at forkas@umd.edu.
Okay, thanks, Dr. Forkas. Peter Nemes, why don't you go next?
Thanks, Amy. So I will try to share. Does this show well on your sides?
Yes.
Okay, perfect.
Except I see 9 slides.
All right, so in the upcoming 40 minutes, I'm just kidding. So welcome everyone to University of Maryland. My lab is, I'm Professor Peter Nemes. My laboratory is focused on the development of ultrasensitive mass spectrometry technologies to enable proteomics and metabolomics studies in neurodevelopmental biology. We are at the interface of chemistry, biochemistry, and also biology. We basically are located right at the intersection of these traditional disciplines. We develop all kinds of analytical instrumentation, primarily focused on mass spectrometry, to enable the study of systems and cell biological processes with down to single cell resolution. My lab also uses various biological systems, and that's what you can see here. We actually work with live animals, including frogs, and we also study behavior. This is, for example, one movie that we've recorded on swimming tadpoles to test for various behavioral phenotypes. So we really link up chemistry with biology and biochemistry. I welcome you to visit our website. You will see a couple of recorded videos there on how we're advancing neuroscience one neuron at a time. All right, now in terms of student outputs, we tend to publish in chemistry and biochemistry and biology journals, as well as interdisciplinary journals. And the students in the group tend to receive a lot of different awards because we're integrating biology and chemistry, and that's a very prominent research direction these days. As I mentioned, we build a lot of instruments ourselves. So these are some of the students who have built these instruments. This is one mass spectrometer that you can see right in the middle of the slide, and then we built things around it. This is another mass spectrometer right in the middle of the slide, and then we built things around it. And then this is another mass spectrometer that we built things around it. And then we have another mass spectrometer that we build things around it so that we can efficiently detect, separate, isolate peptides, proteins, and metabolites from all kinds of single cells and neurons. And then why do we do all this work? Well, first, we've developed proteomic and metabolomic workflows over the last couple of years so that we can now identify literally hundreds and thousands of different kinds of proteins and peptides and metabolites and quantify them. And why do we do all this? Well, because we want to understand basic neurodevelopmental biology better. And so we have a lot of biological topics that we're actively working on. We have discovered molecules that can alter normal self-aid decisions. We study autism spectrum disorder, hearing impairment, human diseases, including the broncho-otorhinal disease and heart diseases. We're studying all kinds of signal induction mechanisms in live developing embryos by integrating high-resolution optical microscopy, mass spectrometry, and molecular biology. We're studying the molecular origin of biological symmetry. So, I have two left hands, it seems, but usually we have left and right hands. So we are understanding how that happens. We're also studying circadian rhythms, vision, stress, and all kinds of impairments that relate to neural processing. My group contains about 8 PhD grad students, a postdoc, 2 undergrads, and 2 high school students. And we're funded by NSF, NIH, various foundations, and professional societies and companies. If you have any questions, here you can see my email address, nemes@umb.edu. And just again, I welcome you to visit our website where you will find a lot more information about our lab. Thank you.
Okay, thank you, Peter. So we're going to do one more. I have one. So Peter, can you unshare? Okay, so I'll just go over this very quickly. Can you see this slide? No. Yes. Okay. So in my research group, we investigate chemical physics and physical chemistry of molecules that have been activated with lasers and optical fields. So we use pulse laser excitation to prepare molecules with extreme and controlled amounts of energy. And we use transient state-resolved spectroscopy to investigate the structure, collision dynamics, and chemical reactivities of these activated molecules. So here is a sample of some of the systems we've looked at recently. UV photodissociation processes of molecules in metastable states that have planetary relevance both on Earth and exoplanets. Collision dynamics of molecules that have been prepared in extreme states near barriers. Optical centrifuge studies to prepare molecules in extreme rotational states with oriented angular momentum. And in all of this, we use state and time resolved product detection. My students learn how to make lasers, how to use lasers, how to build spectrometers, and they learn how quantum mechanics affects the motion and behavior and reactivity of molecules. And so if you're interested in this, then apply to our graduate program and join my group. Okay, so now we have time for a few questions. And one question that was already asked is, what are we going to do about admissions when classes are pass-fail? And I think, Adelina, you're asking about the biochemistry program. and the prerequisite for grades for certain biology grades?
Yeah, my question was just for the program in general.
Yeah.
Mhm.
So when it comes to pass fail, we're just going to have to keep an open mind for this past year, because it's a brave new world for admissions. I think in courses that are going to be relevant for your graduate program, it would be good to have letters from those professors so they could give us a sense of, beyond pass fail. So that's one mechanism. You can also address in your statement of purpose where your strengths lie, because we won't be able to, look at the last year of grades if there aren't letter grades. Okay, are there other questions? Anything about statement of purpose or asking for letters of recommendation?
I have a question, Amy. So if I was applying to the program, what should I put in my letter of recommendation? What should I ask the professors to comment on?
Well, certainly how you know them. And I mean, professors know how to write these letters. I don't think students need to direct the professors. But if there's a special way that you know a professor, say you've done research with them, I mean, professors will know.
Right. It's good if
we
those,
do this a lot.
Yeah, it's good if those letters.
Professors who know you in some way. And
Right.
if you need a third author who maybe you don't know so well, remind them how you know them and how you did in their course and how you were so involved and you were so eager and so curious and dedicated.
Thanks, Amy. That's going to be very helpful.
In a letter, this student is so excited to go to grad school because they're so curious to learn more.
Thanks,
That's
Amy.
what we're looking for.
Great.
Good luck with your applications, Peter.
All right.
Okay. Any other questions? All right. Well, so as far as the format goes.
Sorry, Amy, two things in the chat. One is that a participant asked for Professor Tawari to give his email address.
Okay, I saw that.
And I took the liberty of responding, so it's there in the chat. And Pratush, you might check and just make sure I spelled it correctly. Oh, I think I did. Another participant has asked, can you apply to both biochem and chem? And I would say the answer is yes. You have to apply to them separately. I don't know if that's done very commonly. Amy, you can comment perhaps on that.
Well, you can certainly apply to both. Both applications will come to my committee. So the same people will look at them. You're just going to have to pay a double fee. I would say apply to one, but you could put a note in there that you're not sure which one you want. And we could talk to you more, because I don't think it's hard to switch programs once you're admitted.
It's not hard to switch, and also it's not hard to be a student in one program and work with a faculty member whose research isn't central to that program.
Yeah.
Faculty in the department are not specifically in one program or the other. So faculty can take graduate students who are interested in working with them.
Yeah. Okay. Well, thank you all for coming. Now we're going to turn it over to our graduate student panel, and they're going to talk about graduate student life. So I'm going to thank all the faculty for participating, and I'm going to ask you to leave. Thank you.